Hi, I'm Stuart Tugendreich, Scientific Director for IPA. Today I'm going to talk about the latest release of IPA, which came out at the end of March 2015. In this release, we enhanced our support for mouse RNA Seq data, and in this webinar I'll demonstrate how we used IPA to analyze an RNA Seq dataset deriving from differentiating mouse embryonic stem cells. Before we get started on the use case, let me tell you a little bit about IPA. We are really proud of the fact that there are over 12,000 publications that cite IPA to date. These authors found that IPA was important to understand the biology they are studying. Also, in case you're not completely familiar with all that IPA can do, I want to give you a brief overview of the capabilities of the product. IPA has gene and chemical views that organize all the information that we have gathered from the literature about a particular gene or chemical. These are particularly helpful if you've come across a new gene or chemical in your research that you want to quickly get up to speed about. On those gene views, there's an Isoforms section which displays details about the splice variants for either the human or mouse ortholog of the gene using either the Ensemble or RefSeq gene model. The default is human RefSeq, but you can switch it to whichever human or mouse or Ensemble or RefSeq. IPA has a library of over 650 signaling and metabolic pathways that you can reference and visualize overlaid with your expression data. You can also do prediction of the effects of activation or inhibition of molecules on downstream other nodes in the pathway. You can discover which molecules might be activated or inhibited upstream to explain the expression changes that you saw in your experiment, and also which processes downstream might be activated or inhibited. For example, biological processes like apoptosis or disease processes like cardiomyopathy. The Regulator Effects analysis automatically creates hypotheses for how the activation or inhibition of those upstream regulators could lead to the increase or decrease of expression of target genes in the dataset to subsequently drive particular phenotypic outcomes. IPA has great support for microRNA, including a special target filter which enables you to figure out which microRNAs are known or predicted to target which mRNAs, enabling you to study the system-wide effect of the microRNAs or to focus on key microRNA-mRNA interactions. The microRNA target filter enables you to upload both microRNA and mRNA data into the same table and look at the pairwise interactions with the expression data in context. Bioprofiler helps you understand and focus on how genes or chemicals associate with particular diseases or biological functions. For example, to find the cases where gene knockouts led to increases of a particular phenotype. Lastly, IPA offers a number of means to explore the relationships between genes, proteins, and more. One nice aspect of this set of tools is that you can easily interrogate any such pathway to find out what else is known about that set of molecules. For example, are they known biomarkers, drug targets, or are they in particular canonical pathways, for example? This is all powered by our scientific curation process. We've had PhD level scientists curating findings from the literature for nearly 15 years now and have built up a knowledge base containing over 5 million findings available in IPA. These findings are structured into an ontology with contextual details such as species, specificity, cell types, tissue context, site and type of mutations, direction of change, post-translational modification sites, epigenetic modifications, and experimental methods. These contextual details allow you to find specific relationships such as transcription factor X increases expression of a gene Y in a specific cell type. Okay, so let's talk about what's new in the spring release. I just mentioned there's a new isoform view for mouse. You can also, there's a new, and we enhance the analysis summary tab. I'll go into more details on these in a second. There's a better way of finding genes or chemicals in your data set, and we improve the legibility of nodes on the networks and pathways by the text. You'll see that. And we have 100,000 new findings and 3 new signaling pathways. So the isoform view for mouse I did just mention, but there's pull downs on this gene view in that isoform section, so I can say, I want to see the mouse ensemble view of Pexillin. And if you're coming from a data set RNA Seq data set, you'll see overlaid the expression values from the data set. So this is a 3 observation, as we call them, like three time point data set, and that's the expression changes overlaid on the different, we call these isoform tracks, or the different isoforms that are expressed in the data set are shown here. And we'll go into this in a lot more detail. This is the enhanced analysis summary. So we've make it look nicer, but more importantly, we've added these little graphs that you'll see that are sort of the scatter plot of the p-values for different categories, because sometimes these things represent a category. You know, there's multiple types of cancer underneath in this category, and so we showed the range before, but now we show you the scatter within that range. So to make it more clear, this is like a cardiotoxicity category of toxicity functions, and it shows how there's a number of different p-values. This is the negative exponent, so this is like 10 to the minus 5th, right, or 10 to the minus 8th. And so that's the scatter or the range of p-values, and the little blue line is the median. So you can get a sense of just what it looks like, your data in a glance. And then, of course, you can drill in and look at the detailed heat map to understand that in even better detail. Okay, this in a data set now, you're able to, there's a little funnel, a little filter icon above the symbol column. So in days past, you had to, or months past, you had to like page through the data set to look for a particular gene, or if it's a chemical data set, you'd have to page through for chemicals. But typically, it's a gene data set. You might have many, many thousands of genes. It's hard to find. So now you can type in. You click that little symbol and type it in, and type, find the rows that match your gene name. And it's cool because you actually can. And this is an example of filtering for GATA4. So now you're only showing those rows. But the cool thing is you can also do wildcards. So you can say, RAS star, and that'll find anything that starts with RAS. Or you can even do multiple, type in several gene symbols separated by commas, and you'll find all the rows that match those. And you can do, star RAS star, so that you can find KRAS something, things like that. So wildcarding is very powerful. And just to know, it's not a filtered data set. You're not trying to save this data set. It's really a quick way to find rows of interest to then add to a pathway, or to just look at the expression values for those genes of interest in your data set. This is new. So this is the previous way a network or a pathway might look in IPA. There's a zoomed in little portion here. Now by default, there's a little white border around the font so that you can see it better. I think it's easier to see this much better. So you can turn this off in IPA preferences, but it's on by default, and I think it looks a lot clearer. So there's that. And lastly, for the spring release, we have, as I mentioned, 100,000 new findings and 3 new signaling pathways around autophagy or the phagosome. So please check that out. All right, let's go to the case study, which is around, again, mouse RNA Seq data around cardiomyocyte maturation. Okay, so these were differentiate in vitro, and I'll go into how the design of the experiment. But they're mouse embryonic stem cells that were differentiated in vitro to beating cardiomyocytes over time. The gene expression was measured at the start, like the embryonic stem cells themselves, and then at 2 intermediate stages, and then at a final stage when they're beating. It's a model for toxicology. It's interesting biology to study. And the key things you're going to see is that we see a decrease over time in the embryonic character. The cells are less embryonic and more cardiac nature, like contractility and other functions around becoming cardiomyocytes. The differentiation decreases the cardiomyopathy or other heart-related diseases. You see key upstream regulators are activated that can be tied down to what's happening downstream. And you can actually, one way to use this upstream regulator analysis, another tools like that is to understand what other compounds that actually might help drive the differentiation. So could you improve the assay or this differentiation program by adding compounds? And you can see an example of that. Okay, so the study, which is from a paper from Wamstead, is they took the embryonic stem cells and they added certain activators in vitro to get to what they call a mesodermal stage, and they have markers that they measured of each of those stages, and then they added 3 more activators of differentiation pattern, and they got cardiac precursors, as they called them, and then they waited, and over time you actually see the cardiomyocytes beating. So that the stages are ESC, MES, CP, and CM, and what we did was just ratio, so they're done in biological replicates. What we did was ratio them in this way. So what we call ESC versus mesoderm is really mesoderm, that the second time point ratioed back to the beginning, right? Or what we call ESC versus cardiac precursors is the cardiac precursors ratioed back to the starting. So we're ratioing back to the starting culture in each case and over time, essentially. This was data processed or gotten from the European Nucleotide Archive, and we processed it using the CLC Cancer Research Workbench from this GSE study, and which allowed, it's a very nice tool. You just take FASTQ files and you load it into the Cancer Research Workbench and run the mapping and other what you need to do to map it to the genome, the mouse genome, and then there's a plug-in. You can just connect the data to IPA. You just send the data to IPA. So that's what we did. And we saw really strong differential gene expression in these samples. So so we did filter the data, in CLC Workbench as well as in IPA to meet these criteria. So the absolute fold change of greater than 3, so either fold change plus 3 or better, or -3 or lower. A false discovery rate, which was computed in the Workbench, we less than 0.001, we filtered to that, and an RPKM, or the expression, had to be greater than 10 in at least either the experiment or the control. So in one of the two, you had to have RPKM greater than 10. So we threw out the really low ones that only had, let's say, RPKM 3 and 1 in 2 groups. So we threw out those. And even after doing that fairly stringent cutoff, you see thousands of genes that pass in each case, with the strongest differential expression occurring at the end with the cardiomyocytes relative to the embryonic stem cells, as you might expect. This is a Venn diagram that you can make in IPA. This is a screenshot from IPA showing that there's a couple thousand genes in common that were differentially expressed among all of them. And then there's specific sets of genes that were only expressed. For example, there's a lot of them are only expressed at the cardiomyocyte stage. 2,100 genes, or 2,600 genes that were specific to the mature cardiomyocytes. Okay, so what can we do with this data? We can ask, what are the functional consequences of these changes? How are the biological pathways affected? What upstream is driving this? How does upstream lead to downstream? What happens over time? And what can we learn, importantly, what's going on at the isoform level? There's something interesting there, because that's one of the benefits of RNA Seq data, is you get that level of detail. Okay, so this is a screenshot from IPA of the what we call the downstream effects, which is in the diseases and functions tab of a core analysis. And with this view, as I was saying before, you can really quickly get an overview of what's categorically being increased or decreased, like increased cell movement or increased cardiovascular system development, because almost all of these are orange, or decreased cardiovascular disease, because almost all of these are blue. And of course, that's at a high level. You can click on any of these and drill down and understand in detail which genes are driving those predictions and all that. But underneath the table, underneath this heat map or tree map, you can see a table of the individual squares from here are represented as rows here. So you can see how attachment of cells and migration of endothelial cells are all, these things like that are predicted to be increased with a z-score that's very significant. So just to summarize, you see increased contractility, attachment, differentiation, the heart rate going up, metabolism, carbohydrates going up in this cardiomyocyte. This is, again, the last stage versus the start. And so one thing you can do is do a comparison analysis as well. I didn't really talk about that yet, but you can take your different analyses in IPA and combine them and say, well, what's happening relative one to the other? So this is the three of them, and this is the diseases and functions. Again, you can do this kind of heat map across any of these types of analyses, but let's just focus on the disease and functions. You can see how it's increasing z-score over time, like differentiation is increasing. And the way it was sorted like this was using this little arrow is just drawn on here. There's a little slider in IPA, and you can just say, I'm looking for a case where it's going up from left to right. Or you can slide that up and slide that one down and show decreased over time, or whatever pattern you feel like drawing in with these sliders you can do, and then IPA will find the rows that match that pattern the best. So these are increasing trend over time. If you click on a square, you can see which genes were involved in that prediction. So I'm going to mention this later, but these little fuzzy yellow halo is indicating about the isoforms, that there are multiple isoforms in the data set. Okay, so whereas FOXC1 only had one transcript that was measured, these ones, which are off yellow, had multiple, and we'll go into that. Okay, so let's take like kind of the big overview of all the little pieces of from the heat map that tell you these increases or over time. So these are all increasing. So differentiation, as I mentioned, disease-related, are going down. So these little arrows over here I drew in there to show you, what the patterns. These are increasing over the time course. These things like cardiomyopathy are decreasing over the time course. Some you only see at the very end. They're increasing, but you only see a signal at the very last time point. So the metabolism, polysaccharide. You see contractility and heart rate going up. Migration, the cells are moving around, predicted. There's the change in the cellular dynamics, like using, like microtubules and actin, things like that are changing as the cells progress. So that gives you a kind of big overview of what functionally is happening over time. So we can do the same kind of analysis at the pathway level. So those are functions. This is now canonical pathways. And so you could see how certain pathways activity is predicted to be increased during the time course. And just let's focus on ILK signaling. So that was actually slightly decreased at the beginning maybe, but then it strongly increased in activity prediction over the time. So this is the zoom in of the ILK signaling pathway. You can see lots of different genes in the data set at the final stage. Were upregulated, some are downregulated. And so one thing you can do is query this pathway as to what functions are relevant. There's some that are already here that are drawn into the pathway, like cell motility and how it affects actin polymerization and things like that. There's a focal adhesion theme here, but you can also add functions. So this is a relatively new feature where you can say in the build tool you can grow. So you go to build, then you say grow, and then you say diseases and functions, and it'll automatically compute the statistically relevant diseases and functions for that set of molecules shown on the pathway. And so there's a very strong p-value for development of cardiovascular system for the ILK signaling pathway. So what you can do is just click it and add it to the pathway. And so now you can see how development of cardiovascular system, many of the molecules on this pathway feed into that. And then we can turn on this tool in the overlay menu called the molecule activity predictor, which says, given the expression changes seen on this pathway, what impact would that have on development? So you can see it's increased development of cardiovascular system. And these are a couple of actual findings from so sorry, let me back up. If you want to you can double click on any of these relationships to ask, well, how does
FAC or whatever molecule connect. How do we know it has effect on development of the cardiovascular system? Well, you can double click and you see findings like this. So in mouse, knocking out DSP decreases morphogenesis in mouse heart, or this other mutant mouse has effect on abnormal development of heart and things like that. So you always can get down to the literature to understand how things are connected or why they're connected. So what about isoforms? So I mentioned the yellow halo that you see around. There's many of these have multiple isoforms in the data set. Let's double click. So if you double click on Paxillin, where did I put you? You were somewhere around here. But anyway, you double click on Paxillin and you see, oh, there it is. Double click on Paxillin and you see this little pop-up, which is a summary. And there's a little section called isoform data, which defaults to the right one for your data set. So this is it's a mouse ensemble data set, so that's why it shows mouse ensemble version. And these little rectangles indicate the known isoforms from ensemble from this mouse gene. So there's many of them. Some of them, the white ones, don't have any expression in the data set, but the ones that are colored do have expression. There's 4 of them. And the way these are ordered, left to right, is from largest to smallest transcript. Essentially, these are first we look at the largest protein coding, and then once the ones that don't code for protein, we look at the largest at the nucleotide level. So these are protein coding ones will be at the far left, the largest ones. And these little slashes mean that they didn't pass the expression cutoffs. So they were expressed, but they didn't pass my 3-fold change, that kind of expression cutoff. And then there's a check mark, meaning one of them was chosen as to represent the gene. So that color is what gives Paxillin node the colors, this isoform. But this is definitely a preview. You can't tell that much about it, although you can hover over these to see the name of the isoforms. It's better to click through and see the gene view itself. So this is the Paxillin isoform view in ensemble, as I was mentioning, and here's the expression data overlaid. And you can see how over time several of these are increasing in fold change relative to the starting culture. In fact, and then there's one of them that's not significant enough to pass the cutoffs, but it seems to be decreased in expression relative to the starting culture. And so it's kind of interesting because this is one of these is a different isoform, and one thing we can say is that the Paxillin alpha, this one, occurs in focal adhesions. That's a paper that found that that particular isoform, or the protein expressed from that gene, that transcript, goes to the focal adhesions. Okay, so changing gears a little bit, let's look at the upstream regulators. So again, this is from the comparison analysis and looking at transcription factors only. So upstream regulators can be really any kind of molecule. You can have a microRNA that's an upstream regulator. You can have a compound even. It's something that could drive the changes in expression that you see. This is filtered to transcription factors, and you can see many of them are increasing. Okay. Okay, guys, can you hear me now? From my control panel here, it looks like the sound is fine. My computer is detecting me. Anyway.
Some people can and some people cannot.
Okay. Guys, terribly sorry. It seems like GoToMeeting is having a conniption this morning. I'm just going to keep going because it's hard to solve it for some people. And I think what we're going to do is send this out. We'll record this and we will send this out as a link in the next couple of days. So you'll have, hopefully, I would expect a completely good audio version of this so you can watch yourself afterwards. And again, terribly sorry. Okay. So transcription factors that increase or decrease during this differentiation program. One interesting one is HDAC5 starts out as predicted as increased relative to in the mesoderm cells versus the starting culture, but then decreases over time in predicted activity. So let's take a look at that. There's also compounds, and we'll talk about that in a minute. But So HDAC5, this is the set of molecules from the data set that that are arranged around the predicted transcription factor, so or transcription regulator. And so this actually, just to give you a little understanding of this, the way this works, the coloring works, is that when since HDAC5 is predicted to be activated, it's by the literature it's supposed to turn down the expression of KLF2, and that's why it's blue and an inhibitory edge. So that's consistent with this HDAC5 being activated, is that KLF2 would be downregulated. Likewise, HDAC5 is supposed to increase the expression of twist1 as based on the literature, and it's orange and it's activated meaning that yes, it's twist1 is supposed to be activated or increasing expression by HDAC5, and it is. And then there's conflicts where catenin beta 1 or whatever is supposed to be decreasing expression, yet it's been turned on in the data set, so that's a conflict, and those are shown in yellow. Just as a point of interest, HDAC5 is known to affect the development of cardiomyocyte, so that's kind of cool. And okay, so going to the next slide. Another thing you can do is look at a mechanistic network, which is a way of seeing which regulators are connected to one another. And so HDAC5 has a mechanistic network this is automatically created in IPA when you do the upstream regulator analysis, and you'll see how it's predicted to decrease MyoD1. These are all regulators, so HDAC5 is supposed to decrease MyoD1's activity, and which, etc. So these are different regulators connected together. These control, as a group, 775 data set genes, which would all be connected to these, but which aren't shown just because it's too complicated. But so that's how a mechanistic network that HDAC5 might be regulating. Okay. I mentioned the regulator effects, and again, this is a hypothesis for how a regulator or multiple regulators might drive downstream effects. So this regulator effects network showing how HAN2 and MEF2A might working through these data set molecules by changing their expression leads to this muscle contraction and these other downstream effects. What's kind of cool is there are known cases, so like HAN2 is known from our knowledge base, it's in the knowledge base from the literature, that HAN2 affects the contractility of heart. There is no such connection known in our knowledge base around MEF2. Why is MEF2A, the prediction is MEF2A somehow has to do with contractility of heart, but there's no connection known in our knowledge base. But what's neat, and we curate human, mouse, and rat literature, but if you go to literature, so that's the connection I'm talking about. If you go to the literature, there is a paper from 2005 around zebrafish. So zebrafish is known to, MEF2A is known in zebrafish to drive cardiac contractility, and so that's kind of interesting because now you see it in in a mouse data set, but it was known in a model organism like that, like zebrafish. Turning to that topic of compounds as upstream regulators, again, maybe if you see a compound that's predicted to be activated, this is in IPA, solumetinib is predicted to be quote-unquote activated in this data set. And what that really means is that the expression pattern that's observed in the data set is consistent as if solumetinib were in the system, were applied to these cells. Of course it's not, but the expression data is consistent with that. So that can imply the fact that maybe you could add this compound and drive the differentiation of the cells. So that'd be a kind of experiment you could go to the lab and actually try it. There is one published link to cardiac research of this compound. I mean, it's a very specific thing about cardiac function in a particular mutant situation, but it's just one glimmer of hope that maybe solumetinib would really do that, what we're predicting here. In our knowledge base to connect from a predicted regulator down to the data set molecules. There needs to be literature that supports this regulator being involved in the expression of those 4 targets, if you want to call them targets in the data set. Causal network says, well, let's use the network effects that says maybe B, a different regulator B, works through these pink other regulators and in turn drive the expression of the targets in the data set. So it's finding these higher, more distant connections between regulators. And so it's a very predictive and could find completely novel regulators because, again, like in this little mock-up here, there's one paper, let's say, that talks about B affecting the expression of this one, but there's no other papers that talk about how B might affect this one. It works through a regulator. So you're finding novelty there. And because it can be complicated, there can be a lot of different regulators predicted, we allow you to ask the question, does B have a known connection to some phenotype or something of interest, like another gene, and to help you sort of sort and prioritize the regulators that come out, these Bs, or these upstream master regulators, we call them. And so you can put in a function to score against, for example, and that's what we did exactly here. So this is one network that came out of a causal network analysis showing that the GCNF gene, which we call NR6A1, working through OCT4 or POU5F1 might drive the expression of a number of genes. And there's a paper that's underlying this that says that the gene encoding GCNF plays an important role in the repression of OCT4. I mean, that's that linkage, and it affects somatic cells during early development. And so, again, the scoring, we put in differentiation of cardiomyocytes as an endpoint. So how does any of the regulators have to do with that? And this is the connection that GCNF or NR6A1 has with differentiation of cardiomyocyte. It works through these other potential molecules to drive differentiation. So it's a hypothesis for how this regulator might lead to differentiation. Okay, and this is basically the last topic. Is we have another capability in IPA called My Findings, which is available for additional cost, but it lets you put in your own findings into IPA and use them in your analyses. Maybe there's a topic your company or your institution has really studied in a specific way, your lab, and you want to put those results in IPA and use them in your analyses. You can now do that, and it lets you you can have your own secure repository to go into. Nobody else except on your license sees those findings, and it's automatically used in IPA, and you can control the content in a sense. And so, as an example of this, there's an RNAi screening database out in the internet, and you can think about those kinds of findings, RNAi screening results, as my findings. So the idea of, if you have a gene X, a molecule, that you've increased or decreased with a siRNA screen, for example, and you saw an endpoint like focal adhesion changed, now you have basically a kind of relationship like gene X increases causation of focal adhesion formation. So this is like a typical IPA kind of relationship. It has a direction, it has a type, it's a causation relationship, and it affects some function. So we took this data from this database and loaded them into IPA as my findings, and then scored or ran the analysis again, and this is that same comparison analysis. So what you see is that the final time point, this focal adhesion assay is predicted to be increased. And then everything just works as it should. I mean, you get a regulator effects network using this proprietary or this custom function that we put in, and you can see how these two genes, TAZ and GNAQ, working through these data set molecules could drive the increase of focal adhesion. So again, a connection to focal adhesion that came through like in the ILK signaling pathway. And that's it. So IPA has many uses in toxicology research and general research. You're able to study a very complex situation like developing cardiomyocytes and really understand at a molecular level what's going on and be able to generate hypotheses for how to test those features or those hypotheses in the lab. And you can get that level of molecular detail at the isoform level. And just differentiation predicting is predicted to increase and in turn decrease cardiomyopathy. There's upstream regulators that came out, that including compounds that might help drive the differentiation. That's what we can do with this kind of data in IPA. And I just wanted to add that the advanced analytics, which has 3 components currently, can be added to your IPA license if you don't already have it. So there's a special, and there's a special running, so you can share the benefits with your friend. You can get 2 licenses for the price of 1, or you can get 25% off if you buy before April 30th. And again, so the 3 features are causal network analysis, I showed you that, bioprofiler, I mentioned that advanced search tool, and then finally you can do relationship exports. You can export the relationships out of IPA networks or pathways as part of this package. And you can just contact your account manager or the marketing at rwc@qiagen.com to order. And you should act quickly because it's 2 weeks left on this promotion. So again, thanks for your time. And so I guess we might have questions, and let's take a look at those.
Thank you, Stuart. We have some questions here. Would IPA use KEGG database for metabolic pathways again in near future?
Not in the near future. Yeah, so KEGG are still available as gene lists in IPA, but the pathways themselves are not part of IPA. Go ahead.
What is the reference for cardiomyocyte data?
Right, so let me shoot back there really quick. It's Wamstead et al., Wamstead J.A. et al. From Cell in 2012, and if you just go to just do a Google search for GSE 47948, that will find you right to that geodata set, and that references the paper as well. So that's the data. And we did put these into this data set that I showed. We put into IPA as an example analysis. There's a folder in IPA that has a bunch of different examples, and this is now one of them, so you can find it there.
This was a question by Suraj. What do dotted lines mean in the previous slide?
Oh, okay. Yeah, dotted lines. So the dashed or dotted lines indicate an interaction of some sort, but it's not a direct interaction. In other words, it's not a binding kind of relationship. There's no physical interaction, and especially in every single case of a case where there's genes in a function like that, there's never going to be a direct line. There's nothing. You can't physically touch differentiation, right? It's a concept. But in networks like, let me see, okay, like this one, this means a direct. There's a binding or physical relationship, whereas these are indirect. In fact, it's possible there are other molecules even between those two, but GSK3 has an effect on I don't know what, something down here. Yeah, so it's direct versus indirect interactions.
Is there an annotation key for the networks? Can we find it online or on software manual?
Yeah, there are. In the help manual, there's a description of the shapes. What circles mean, what triangles mean, what the lines mean. Yeah, so there's a legend available in the help.
Is regulator effect and upstream effect only use knowledge-based networks, or does it also do some networking techniques to predict the possible regulatory networks?
I mean, it uses the knowledge. It uses their algorithms that underlie their algorithms that underlie the results, right? I mean, there's a z-score calculation. There's a journal article that we published in Bioinformatics in early 2014 that goes into the mathematics behind these kinds of calculations. So there's an algorithm that's run, but the content that powers those algorithms is from the knowledge base. It's from the literature. It's not like we say, well, if there's something that's connected with 50 other things, then we're going to make some assumption about that gene or some things like that. There's nothing inference like that. It's using literal connections that come from literature.
Is isoform view available for human genes?
Yeah, absolutely. So, I mean, let me just go quickly, go to IPA itself and just open a gene. I don't know, TGF beta 1. So I'm jumping to a gene view, and so I wanted to show you how that looks. There's by default, like if you don't have data, if you just come into IPA and you're looking up a gene, by default the gene view is going to show you the human isoform, okay, from RefSeq. So the default state of IPA's gene views is actually human RefSeq. But because this data set that I just launched this gene view from was a mouse one, an ensemble, it knows to go to mouse ensemble. But you can override that and say, I want to see human, and I want to see RefSeq. So yeah, so you have those 4 choices: human, mouse, ensemble, RefSeq. And again, the default is human RefSeq. I'll just switch it back. And again, when because you had this came out of a data set, there are those data.
In this example, why don't you try some comparison in IPA? For example, compare CM with CP, compare CM with MES, and compare CP with MES.
Yeah, I thought about it. It just seemed more straightforward to compare it back to the starting culture. But definitely, and in fact, again, the data is in the example analysis, so you're free to take that data and ratio it yourself. I think you could do that. Yeah, ratio it yourself to find how, how does the CP differ from the CM. So you absolutely can do any of those ratios that you'd like. I just did this. We just did it this way for just to keep it clear.
If IPA finding sources are not from Ingenuity expert findings, for example, from gene ontology or mouse genome informatics, any active and inhibited relationship between 2 molecules will be showed too. I ask this question because some database, for example, they don't curate the active and inhibit relation.
Right. So I think what the question is really asking is, are all relationships directional? Like, do we have increases or a decreases effect for every relationship? And that's no. I mean, in cases where they come from databases that don't capture that, we can't have that. But we try our very best to get directional information. This increases that is much more valuable, as you can see, than just this affects that. So we try to go, when we go to the literature, we get that information, and we try as much as we can to do it. Yeah, and in some cases you can, we try to get it as much as we can.
How do you get value for individual fold change for MES, CPCM? Normally after analysis in, for example, R, you get a comparison with only 1 fold change value.
I don't quite understand the question. I mean, so let me just describe what we did, and then maybe that will answer your question. So what you got out of the CLC software is essentially intensity information, or like RPKM counts for each individual sample, right? So like for the embryonic stem cells, you get an RPKM count or value for each transcript for each gene, and then that gives you many 100,000 rows or whatever. It's many thousands of rows. You then have that same kind of RPKM count for the mesoderm. Then in the workbench, you say, I want to create an experiment between the mesoderm and the embryonic stem cells, and it calculates the ratios or fold changes between those two, and calculates an FDR and all these other values about how that, the significance of that change given the samples you have. But so you have all the basically you have all the RPKM counts, and you just ratio them. And then we do the same thing for embryonic stem cells to CP, and that gives you all the fold changes. I hope that answered your question.
How does IPA discriminate read counts for overlapping regions in isoform? Is there an analysis that needs to be done before importing to IPA, such as CR as you used? For example, does Partake do it, or is it IPA's algorithm?
Oh, I see. No, I mean, all IPA is kind of agnostic to the upstream processing. IPA loves fold changes and p-values and ratios, from pre-processed from some other tool, whether that's CLC or whether that's some other tool, or whether it's something you do in Excel, or whether it's something you homegrown algorithm, whatever. IPA pulls in the fold changes and p-values and things like that. And so, so we did all that upstream processing in the CLC genomics workbench or cancer research workbench, and then just pushed it into IPA as fold changes and p-values and things like that.
How deep were these sample sequenced to see isoform variants? And does the software need a minimum number to make predictions? RPK more than 10 used in this example?
Yeah, I can't remember off the top of my head now what the depth of the sequencing was. You can look that up in that GSE reference that I mentioned earlier. I mean, IPA is sort of, again, agnostic to what you do upstream. If you don't have deep enough reads and your upstream software says, you know, I can't call that not variant, but I can't call that transcript. I don't have enough data to make that determination, and maybe it gives it a p-value of 0.5 or something for that transcript. That's what the software upstream might do. IPA, then when you bring in an IPA, you just filter it away and say, I don't care about things that have a p-value that's greater than 0.01, or I don't want to look at anything that has an RPKM less than 1 or 10 or what have you. So you do that filtering in IPA. If you bring in those values, if you bring in p-values into IPA, if you bring in RPKM, you can do the filtering in IPA. The algorithms, once they pass your cutoff in IPA, they are assumed to be robust, you know, fold changes. If you decide that 0.0001 is your cutoff fold change, well, then it's going to treat everything as if it's true, you know, that passed. Or if you do it at 0.01, same thing. It's just going to take everything you let through the analysis and then analyze it as if they're all believable. So you have to set your cutoff, you know, properly.
So Kerry Nicholas is clarifying his question when he asked how do you get value per individual fold change for MES CPCM. What he wants to say is that if I'm uploading my analyzed data to IPA with log2 fold change, etc., I will only have one value. Are you saying IPA can do an analysis of raw counts?
Again, I'm not quite sure what you mean, but I think you're saying that because there's different transcripts for one gene, there's multiple fold changes for one gene because there are multiple transcripts. So if you bring in ensemble IDs that are transcript level, let's say there might be 5 transcripts for gene X, or in this case, let's go to this one. There are, in ensemble view, looking at your screen, in the mouse isoform from ensemble, if you process your data in the ensemble gene model, you could have up to 3 different fold changes for one gene, for the TGFP1. In this example, the data set only had one fold change for that gene. This is each a different time point, right? So just looking at the first one, there's only one fold change for mesoderm, whatever. And but you could have up to 3 because there's 3 known transcripts. So it's all, again, it's in the upstream processing, how you process it. If you bring in multiple transcripts for one gene, IPA will represent that here as shown.
Does bioprofiler function cost extra?
If you don't already have bioprofiler or advanced analytics as part of your license, if you don't already have it, it's an extra charge, and that's what this promotion I mentioned was about. So there's, up until the end of the month, there's a discount on what we call advanced analytics, which includes bioprofiler, causal network analysis, and relationship export. Those 3 features are packaged under what we call advanced analytics, and you should upgrade your license to include those. Yeah.
Do you have tools to study ink RNAs?
Yeah. So long non-coding RNAs are essentially treated like any other gene in IPA. I mean, it's an RNA that doesn't code for protein, and that's not a problem. And so if you have long non-coding RNA data in your data set, it'll map and it'll just be treated like a gene, which is, I think, fair. If there's a function known about the long non-coding RNA, you could see it in a network and what have you. I think it's fair to say we don't have as much. We need to get more long non-coding RNA content, like the function of long non-coding RNAs. We have some, but there are newly pieces of information about long non-coding RNAs that could be better. So we're capturing that from the literature.
So on to the next question from Elena. Can you explain how toxicology list predictions are formed? What does it mean if a sample has a higher log value for cardiomyopathy?
Those toxicity lists were curated by a toxicologist, panel of toxicologists. So it'd be hard to give you a specific example without going into detail, but it's increased genes in those lists could lead to increase of those endpoints. So that's all I can say about it right now.
How did you create the comparative heat map between different cells?
Okay, so I can show you that really, really quickly. I know we're out of time here, but if you guys are patient, I don't mind staying a few more minutes. So you just go, after you, so you run your analyses in IPA, and if you happen to have them all in the same data set, so you've uploaded, let's say in this example, we've actually concatenated sort of those 3 data sets into one Excel file, right? That's the kind of, think about it like that. Like there's 3 conditions in one file. Then they actually come out as a package of a folder of analyses in IPA. Then it's really easy to just do, file, new comparison analysis, and you can easily pick, your 3, and you just add them there, and you just view comparison. But they don't have to be. I just want to emphasize, they don't have to be in the same physical data set. You can pick them from any place. I could look at, West Nile virus and its effect. How does it compare to cardiomyocyte differentiation? So you can pick them from different sources, but it's really convenient if you happen to upload them together. But the bottom line is you have to create a comparison analysis, and then you get the heat maps, like I just showed you.
I'm referring to the comparison of the group comparison.
Oh, sorry. Comparison of the group comparisons. Comparison of the group comparisons. Do you mean the Venn diagram? I'm not sure what you mean by that. If you're talking about let's just pretend you're talking about the Venn diagram. You just go to new compare, and that is the Venn diagram. So that's where you can choose, different analyses and add them to the Venn diagram. So that's the difference between compare and comparison, is that compare is more like the Venn diagram, and you're comparing the genes within an analysis, whereas comparison analysis is the heat map. I hope that was what you're asking. Maybe not.
Carrie, just want to mention, if you have specific question, you can also write to us at marketing-rwc@qiagen.com, and we will answer all your questions, and we will have one of the salesperson call you. On to the next question. If you upload data that represents the comparison of 2 groups with fold change, how are you making additional comparison with heat maps? That's the same question.
I think what you're asking is, if I had 2 conditions, and those conditions represent, so an analysis in IPA really operates at the level of, again, fold changes. So I have X, I'm comparing tumor to normal, okay, let's say, and I have a ratio of tumor to normal. Now that gives me fold changes. That's one analysis, okay. Now if I have another analysis that's like a different tumor type to normal, that would be that could be a second analysis. So I can get, those have fold changes, right? Now I can do an analysis of those, and I can do a comparison analysis of those 2 analyses. So I can say, how does analysis 1 compare to analysis 2? So I'm basically asking what effects is, the tumor happening in the tumor versus the normal versus this other tumor versus normal? How do those 2 compare? It's just it's really just lining them up side by side. It's not like explicitly doing some sort of ratio. It's just saying what are the upstream regulators for the first analysis, what are the upstream regulators for the second analysis, and how do they compare? I can explicitly look at them because they're in a heat map. I can compare the 2. If you want to then do something like show me tumor 1 versus tumor 2 instead of versus normal, you again would have to do that upstream of IPA. You'd have to create those ratios or fold changes of tumor 1 versus tumor 2, and then you could do that as an analysis, and that's a new analysis, and then that's really looking at the fold changes, how tumor 1 versus tumor 2. I hope that answers your question. It's sometimes hard. Okay.
Can you talk a little bit about what type of input data we should provide to get a differential expression analysis down to the isoform level? Should we input the read report for each condition and do a comparison analysis, or provide the list with FC and p-val already? If the second option is the favorite, then what is your favorite RNA Seq analysis algorithm? Cufflinks, CuffDiff, or DC, for instance?
Okay, yeah, so it is the second. Again, really you want to upload into IPA the already processed fold changes, p-values that you do upstream in some other tool. I can't really recommend any particular algorithm. That's sort of up to you, or look in the literature to see what the best one is. I like, as in this example, I find it really easy to use the CLC cancer research workbench software, and it has its own algorithm. I think it's EdgeR, I think. So yeah, I can't really recommend one or another at this point.
Can IPA use splice form restriction in network analysis? For example, exclude, include only a subset of isoforms during analysis?
I'm not sure what you mean by splice form restriction, but any kind of, I mean, the only kind of filters you can apply as far as isoforms, there's really no filter that you can apply in IPA about isoforms. You can say, I know the fold change has to be at least 2 or 5 or whatever, which operates on all the isoforms in the entire data set, but there's no way to say only, hey, only show me protein coding isoforms or something like that. You can do that upstream in whatever software you use, but IPA doesn't operate at that level.
Are there plans to increase hierarchical clustering capabilities in comparison analysis? I have 144 single-cell RNA Seq data and can only cluster compare 20 at a time. I have developed a manual algorithm, which is tedious.
Now, at this point, we don't have plans to increase that limit from 20 to something larger. So at this point, we don't have plans to do that. We do have, in the future, we're working on having a gene level kind of heat map. That's something we're aiming towards in the future, where you can see, for a given, in the comparison analysis, show me a heat map of the genes that are relevant to this upstream regulator, as well as the heat map of the upstream regulators themselves. But we don't plan to increase that limit at this point of 20.
A general question, does IPA only work on species of human, mouse, and rat? Because I see they are the species-specific identifiers supported in IPA. Could it work on plants for pathway analysis like corn?
So I mean, IPA is geared towards human, mouse, and rat, and we've curated the literature around those 3 species. Not to say people don't use IPA for all kinds of species, and in fact, if you go to our website, you can type plants or something. You can get a search result. One of the search results is citations. So you can see there are places that people name plant, but there are also people that are corn. You can type and see what people have used IPA for that in the past. So again, it's a little bit corn oil. But anyway, let me just put it this way: people have used IPA for other species. You just have to provide IPA the human, mouse, or rat equivalent gene. So what they do is you go to a place like Ensemble and go to I think it's Biomart, and you can convert identifiers and say, I have these corn genes. What's the closest human, mouse, or rat equivalent? And then you just basically fool IPA into thinking you're actually doing an analysis of human, mouse, or rat genes, and then you can get the result. Of course, we're not going to have findings around plant stalk length and things like that might be relevant to plant, but you can at least get a sense of what's happening, because cells are cells in a lot of ways, and you can learn a lot about what's going on in your plants based on what you see in IPA. So, yeah.
How do you export the Venn diagrams?
Unfortunately, you can't at this point export them. You just take a screenshot on your computer. Sorry about that at this point.
How do you make heat maps for the disease functions, and how do you export them?
So I think there's 2 kinds of heat maps in IPA, right? There is this kind of heat map, the disease and function heat map, which is this tree map, which is basically a hierarchical heat map. If you're talking about that heat map, there's that heat map, and then there's the comparison analysis heat map. And either one of them, unfortunately, at this point, you can't export them as an image, and it's something we would like to change. But you can take a screenshot of this image. You can move things around, make it bigger, and it's interactive, which is nice, right? So you can also do things like make the size of the squares dependent on the z-score. So now it's only showing larger ones have larger z-scores. You can size them by the number of genes, so the bigger ones in this case would have more numbers of genes in them. So there are a lot of things you can do. You can look at the p-value instead of the z-score. Lots of things you can do interactively. You can't export it at this point except take a screenshot. Again, you can also drill down and understand the details. So.
When you filter the differential expressed genes, why do you use FPKM, not the original number of mapped reads to the gene?
Yeah, I just felt it better to use the normalized sort of based up, normalized by length of the gene. You can do it either way. I mean, this is not rock solid. You have to do it this way or it's wrong kind of thing. It's just the way we did it.
What p-value minimum would you consider to be the lowest criteria for further analysis?
Well, I don't know. I mean, in general, people have said, 0.05 is the highest p-value or the worst p-value that you'd want to use for certain things, but there's really no law about any of this stuff. It's best practices. In general, in IP8, the number of genes we put into the analysis needs to be in a kind of range. Needs to be in a kind of range. Like you don't want to put in 3 genes for an analysis, right? It's just too few to really make any biological sense out of really just 2 or 3 genes. Also, you don't want to put in 10,000 genes, so because then you're like talking about the whole genome or half the genome or a quarter. So we tend to like to put in a few hundred to a few thousand genes for the analysis. So we often like change the fold change or the p-value cutoffs a little bit to make sure that that happens. Like in this case, I usually use less of a fold change than 3, but there's so many genes that were significant that came through if you used a fold change of 2. So that was sort of the thinking.
So what is the maximum number of gene transcripts in your data set that can be included in analysis?
Right. I think it's 8,000 genes is the max you can do in one analysis.
The next question is, what's the version of ensemble gene ID and transcript ID can be identified by IPA? For example, release 77 or 78?
Oh, that question just came up, but I can't remember the answer. We'll send an email back to you. I've forgotten that.
And we have another question is infected versus uninfected ratio of treated versus untreated, what they must mean?
Basically, those are 2 conditions. You have expression values for one condition, like treated with something. I'm not sure what you mean, or untreated is the mock condition, or treated with mock conditions. And then those are the way people generally set up gene expression experiments, is something versus something, and it's that comparison. It's a differential expression that's always what you really want to analyze. That answers your question. So maybe we've gone way over time. I'm impressed that people stayed on so long. I really appreciate that. So thanks so much, and have a great day.
Thank you, everyone. Anyone who has specific questions can always write to us at marketing-rwc@Kaizen.com, or you can call us at our customer support line. Thank you so very much for joining us. Bye. Have a good day.
Bye-bye.